All right, good afternoon, everyone. Looks like everyone's here. Okay, for those of you who turned in your homework, let's go over the answers. It seemed like there were some problems with the exception of a couple of you. You did pretty well. So let's go over homework number 10. Here it is right here. So I sent this newer format. It's a little bit clearer. CBCs 1 through 5 and questions here. If it's reportable, tell me what the problem is and then the corrective action. Okay, so number 1 turned out to be normal, no flags, and so that's just a normal CBC. Reportable, yes. Problem, normal results and no corrective action. Number 2, if you look at number 2, you take a look at the results. Okay, the whites are low, the reds are low, the H and H rule of threes passed. Okay, I know 12 times 3 is 36 and 37 is not 36, but the tolerance is plus or minus 3. That was in the lecture. So rule of threes passed. The MCV is ridiculously high. Okay, so something's going on. A blob went through the flow cell. A blob went through the flow cell. Okay, MCHC. Okay, so people say MCHC, made comments on the MCHC. I had mentioned that the MCHC to me is not clinically significant, so I don't care if the MCHC is high or low or whatever. So because it doesn't tell me anything. The only thing it'll tell you is if it's low, there's no hemoglobin in the cell, so you get hypochromic. We're not looking at the cells right now. We're looking at the CBC. Okay, so things like MCH and RDW, we're not looking at the cells right now. So whether it's hypochromic or whether there's an ESO, I really don't care. Right now, at this point, we're trying to look at to see if the CBC is acceptable at this point from looking at the printout. Okay, MCHC is fine. MCHC failed. Remember, 32 to 36, definitely not 36. So there's something going on here. And platelets are low. So 3 cell lines. What are the 3 cell lines that are in the blood? The whites, the reds, and the platelets. So whites, reds, and the platelets. So is this aplastic anemia? No, it's not aplastic anemia because the H and H is fine. I mean, if this was aplastic anemia, you'd have a hemoglobin of 4 and a hematocrit of about 12. H and H is fine. This is livable. The patient's fine. So what this is, all the cell lines are low. This is a clotted sample. Okay, so there's fibrin. Remember, fibrin is the magnet for everything. It sucked up the whites, it sucked up the reds, and it sucked up the platelets. So this is a clotted sample. Okay, and it's not anemia. Okay, so that clot went through the flow cell. That the MCV should have told you that. A giant glob went through the flow cell. Normal for MCV is 80 to 100. 120, okay, a glob went through the flow cell. MCHC failed. Usually on the CBCs that failed, the MCHC is not 32 to 36. Okay, so this is a clotted sample. Redraw the patient. That's a corrective action. Okay, the third one. Okay, again, look at the cell lines. Whites are fine. The reds are ridiculously low. Look at that, 0.8. That's probably not compatible with life. The H and H, okay, here again, H and H, 14 times 3 is 42, but 41, is that tolerable? It's within the tolerable limits. 14 times 3 is 42, so 41 is okay. Oh my God, look at that MCV. A glob went through the flow cell, 130. Remember, normal for MCV is 80 to 100. 130, something huge went through the flow cell. What is it? Okay. MCHC, who cares? MCHC failed because, not because of the H and H, but because of the MCV. RDW, who cares? Platelet counts are normal. So what's unusually low here? It's the reds. So this one is cold agglutinins. Okay, cold agglutinins is when the red cells stick to each other. Where did they go? They went here. Okay, they went here. They went to the MCV. So the reds are low. So this one is cold agglutinins. All right. So cold agglutinins and the corrective action is pre-warming. Number 4, okay, take a look at the cell lines again. Whites are fine, reds are fine, H and H is fine. 12 times 3 is 36. Again, 37 is fine. MCV is huge, okay? 80 to 100, well, 135 is really, really high. So again, a glob went through the flow cell. Okay, whites and reds are fine. So it's not whites or reds. MCHC, who cares? MCHC, is it 32 to 36? No, it's 50. So this one is not reportable. Where did your platelets go? Where did your platelets go? It went here. Okay, so this one is platelet clumping. This is platelet clumping. I went over this in class. So this one, corrective action, is redraw it in citrate. Okay, number 5. Again, take a look at the cell lines. Whites are fine. Reds are a little bit low, but it's tolerable. It's not like this kind of low, okay, or this kind of low. 3 is fine. It's just abnormally low. Maybe it's a viral infection, but it's still a viral infection is reportable. Rule of threes. Take a look at the rule of threes. What's 22 times 3 is 66. This is incredibly high. So what kind of things would cause this hemoglobin to be that ridiculously high? Probably lipemia, probably high protein, probably icterus. Okay, keep that in mind. MCV, probably the globulins in the lipemia. MCHC, who cares? MCHC will fail. 32 to 36. So MCHC is out. And your platelets are fine. So whites are fine, reds are fine, and platelets are fine. MCV is not. So what's going on here? What's incredibly unusual here? The rule of threes, okay? Why did that hemoglobin read so high? It's because of the spectrophotometer in the machine. So what is the problem? It's not cold agglutinins. Your reds would have been low, like in here. It's not cold agglutinins, which some of you said. This one is lipemic icteric. So in this case, what you would do is a saline replacement. Okay. So this is recorded. Please review the recording after the lecture. So that's lecture 10. All right, that's lecture 10. So tomorrow, at the end of this lecture, I'm going to give you an abnormal RBC quiz. We went over all the possible cells that you're going to be quizzed on later today, and hopefully you're reviewing those. You'll do the test. I'll send it out to you by email, and you'll just return it to me by email. All right. And then after my lectures today, I'm going to go over, review your quizzes for tomorrow. And then I'm going to go over, I went over the lecture portion of your exam 2 last Wednesday. So I'm just going to review the not the multiple choice, but the matching portion of your exam 2 after the exam. So I'm going to review the quizzes, and then I'm going to review the matching portion, because you haven't had matching as part of your exam 2 so far. So matching on exam 2, and then I just went over a homework 10. You're gonna, on the case studies, you're gonna be seeing probably questions like this. You know, take a look at the cell lines. If it's everything, you know, just make sense of everything. And if you notice, the MCVs on all of them, except for number 1, which is the normal one, were incredibly high. So try to figure out what's going through the flow cell. Big things could be cold agglutinins, big things can be platelet clumps, big things can be fibrin strands that have whites, reds, and platelets stuck to it. Okay, that's what you need to be thinking about when you're looking at these CBCs with artifacts. Okay, are there any questions about this, about the homework? Is it clear now? Okay, so one thing about asking questions, only one person regarding the homework 10 had asked the question, requested clarification, and I responded to it, and then I sent out an amended homework number 10. I had asked, you know, throughout the semester, if you have any questions, ask questions. Well, this person asked questions and he got all CBCs correct. Okay, if you're not asking questions, then it's only on you if you get the problems wrong. It's only on you. I mean, if you're going to sit there and not say anything, not make any comments, or what about this, or I thought it was this, or can you clarify something? If you don't let me know, I won't clarify it for you. But everything that I mentioned on these 5 CBCs, I mentioned in class. Okay. All right. So let's move on to the first lecture. Okay, so the first lecture is lymphocytes. Lymphocytes is kind of a difficult lecture. It's kind of complicated because there's different types of lymphocytes. There's antigen presenting cell, there's T lymphocytes, there's lymphocytes that are not supposed to, lymphocytes are technically not supposed to be granulocytes. Your granulocytes are classically the ones in the myelocytic series, the myeloblasts, promyelocyte, myelocyte metas, etc. Those are the granulocytes, the eosinophils, the basophils. The monocytes actually have granules, and I'm going to talk about in the next lecture after this. Monocytes, they have granules, but they're not considered granulocytes. Okay. Don't ask me that I got into a debate with somebody, but there are granules in monocytes. Lymphocytes, on the other hand, are not supposed to have granules, but it turns out, you'll see in the middle of this lecture that the T killer cells have granules, okay, that are used to break down whatever it is it's attacking or attaching to. So this is lymphocytes. Okay, lymphocyte function. 3 major types of lymphocytes. Those are the T cells and the B cells and the natural killer cells, the NK cells. But the main ones that I'll be talking about in this lecture are T cells and B cells. Okay, so the T cells or the thymus cells or the B cells or the bursa-derived cells. Bursa means it actually derived from the bursa of Fabricius, which is an organ on a bird, but it's B cells. There's T and B cells. These are the major cellular components of the immune system. You're going to be asked that. What are the major cellular components of the immune system? And you need to answer T cells and B cells. So to distinguish the two, T cells are involved with cell-mediated immunity. Okay, so what does that mean? Whenever you have a virus infection, a fungus infection, a protozoan infection, like E. histolytica or E. coli or whatever, or if you have cancer cells, or transplant rejection. For example, I don't know if you've heard of graft versus host disease. Okay, I'll be talking about that. You'll be talking about that in blood bank. But graft versus host disease, if you get a transplant, an organ from another human being, you may reject that. You may reject that organ or that donation, and what causes the rejection are T cells. So that's cell-mediated immunity. It's recognizing these things that are in your body, the funguses, the viruses, etc. That's cell-mediated immunity when your body reacts to those. And there's also cells called the T helper cells, and the T helper cells are needed to activate the B cells and macrophages. Okay, the B cells are the other cells. Okay, B cells, on the other hand, are primarily responsible for what's called humoral immunity. So what happens in humoral immunity, you're actually producing antibodies. Okay, humor means cell-free fluid, but the important thing to know that humoral immunity is that you're producing antibodies. You're not really producing antibodies in cell-mediated immunity, but if I say antibody production, excuse me, I'm talking about humoral immunity. Okay, and that's B cells. And so the thymus is located here between the upper lobes of the lung. Upper lobes of the lung. That's where the thymus is located. Okay, so the function of T cells and B cells is to recognize special non-self or foreign antigens. Okay, so whenever you have particles or anything that enters your body that's foreign. Your T cells and B cells will recognize that as it doesn't belong in your body. So it's going to say, no, I'm going to react to it and I'm going to do something about it. Okay. Don't get that confused with alloantibodies. I know that's in blood bank. We talked about that with the anti-D and anti-Cal, etc. That's not allo immunity. This is now we're talking about recognizing self versus non-self. Okay, so once these cells, the T cells and the B cells, have identified the invader, okay, the particles that have entered your body, the cells would generate specific responses. Okay, that's what I mean. Your body's going to say these things, these invaders, if you will, or particles, if you will, do not belong in your body. So I'm gonna do something about it. Okay, so that there has to be, your body's going to respond. Okay, so what happens is the B cells, these, when I say B cells or T cells, these are actually lymphocytes. Okay, if we look at a lymphocyte, we don't know if it's a T cell or B cell. If it produces a plasma cell, then it's a B cell. Plasma cell is a cell that actually makes the antibodies. Okay, plasma cells are B cells that actually makes the antibody. Okay, it responds to pathogens by producing large quantities of antibodies. So make sure you know that plasma cells make antibodies and they come from the B cells. Okay, and then in response to pathogens, some T cells called T helper cells will also produce cytokines. So T cells will activate the B cells. The T helper cells are needed to activate B cells, but another function of T helper cells is to produce cytokines. Okay, cytokines are not cells. What they are proteins. They're actually proteins that help in the communication of, they're actually, if you can picture like an air traffic controller will say, yes, you can land, no, you can't land because this plane is landing, you need to hold off for a little bit, or this, you need to go to this runway, or you need to go to that runway. Cytokines kind of like directs traffic, okay? And that's what T helper cells will produce, cytokines. Another form of T cell are called cytotoxic T cells, And these cells produce toxic granules. There's that granule thing again, okay? Generally, lymphocytes do not have granules, but cytotoxic T cells have granules that contain enzymes. And it's the enzymes that will induce death of the pathogen infected cell. So say, for example, a virus, for example, when you have a cold. So the reason why you get sick is that virus, it's not the virus that's making you sick, it's what it does in conjunction with the cell that infected. You have infected cells that are infected with, you have cells infected with viruses that have reproduced. You don't get well because they reproduce, destroy the cell, and move on to other cells, and I'll explain that process to you later. So the viruses and these infected cells are what makes you sick. So what's the cytotoxic T cell is to identify those cells that are infected, the pathogen infected cells. Okay, the pathogen infected cells. Usually it's a virus infected cell, or it could be a bacteria infected cell. But it's up to the cytotoxic T cell to identify these pathogen infected cells and get rid of them. Okay. It kind of acts in the same way as macrophages. Macrophages will go around, they float around, they swim around in your body, and they look for junk that don't belong in your body. Okay, like for example, when red cells get, when they're at their life expectancy and they get destroyed, the stroma of the red cells, they just clean up. Okay, macrophages are like the garbage trucks. They see garbage in your blood and they'll just clean it up. But in this one, cytotoxic T cells, they are looking for infected cells. Okay, so they hunt around, identify a pathogen infected cell, and just clean it up. And I'll go through that process later. So cytotoxic T cells are really important. So cytokines. by definition, are low molecular weight soluble proteins. These are proteins produced in response to an antigen produced by, remember, the T helper cell. Okay, function as a chemical messenger. If you, like I said, if you, it sends messages to regulate the immune response function. For example, if you get a cut or if you have an infection, you have a whole bunch of cells who don't have their marching orders, or they don't have orders to go where they need to go. It's the cytokines that will give those marching orders, or give those orders. You need to go to this part of the body, and then you need to start. It'll tell the cells where to deploy. Okay, that's what the cytokines will do. Chemical messenger to regulate the immune response function. So something's happening in your body, the cytokines will be activated. Okay, cytokines is a category of signaling molecules and used extensively in intercellular communication. So communication is a big aspect of the cytokines, and intercellular means between the cells. Okay, so the cells will know where to go, and it's the cytokines that gives them direction. So the effects of cytokines, there are main effects of cytokines, and you'll be asked this. They're important in increasing inflammation. They increase vasodilation. Inflammation is your body's response to an infection. Say you get a cut. If you get a cut, you notice it'll turn red and it'll start swelling up. That's inflammation. Okay, that's not necessarily infection yet. It's inflammation. Inflammation is your body's your initial response to something like a cut. Okay, increased vasodilation. You want vasodilation because once your blood vessels the blood vessels increase in size, then that facilitates the movement of the cells to that location. So there's vasodilation as opposed to vasoconstriction. So vasodilation facilitates the movement of cells or blood to the location of the injury. Coagulation, it has coagulation properties because you want to wall off the injury. You have a site of injury, you have a cut, there's going to be coagulation going on, and your body wants to wall off the area of their injury. If it can coagulate, then it creates a barrier. And then there's another process called transmigration, and it's called diapedesis. Diapedesis, and there's a pretty good diagram coming up. It's the passage of blood cells through the intact cells of the capillaries, typically accompanying inflammation. So what happens is the cells, on their way to being mobilized to the site of injury, they'll take shortcuts. And the way they take a shortcut is by going through the intact cells of the capillary. They won't necessarily follow the road to the site of injury. They'll actually take a shortcut and go through the wall of the capillary, and it'll be able to do that. And then another sign of inflammation is elevated fever, the temperature of the cells. If you recall, if you cut yourself or you're infected, it feels warm. So you have elevated fever, and that's the temperature of the tissues. Specifically, that's caused by pyrogens, like it generates like fire. So elevated fever is caused by pyrogens, but that's part of the inflammatory process. So cytokines are nothing more than chemical messengers. They're proteins, not cells, and it mediates directing traffic. Okay, and it's starting the process of defense. So something happens to your body, you get a cut, the cytokines get activated, it tells your cells where to go, where the site of injury is. Acute inflammation is the immediate short-term localized protective response to tissue injury, okay? And cytokines is actively involved with that. Inflammatory cytokines initially prepare, mediate, and control the body's response to local injury. That's what I've been saying for the past 8 or so minutes. We want this. We want cytokines to cause inflammation. So you get an inflammatory response. If you get that, we want this. It's necessary for life because your body has to respond to whatever is harming your body. So what cytokines do, they stimulate chemotaxis. Okay, like I said, it helps the cells get deployed to the site of injury. Phagocytes through opsonizing bacterial surfaces. So you know what opsonizing is, right? It makes bacteria, say for example, you have a bacterial infection. Well, cytokines will opsonize bacteria. It'll make the bacteria either more visible or more, I hate to use this word because when I was taking immunology, she said it makes bacteria more delectable. Well, I'm not going to use delectable. It makes it more appetizing. Say, for example, you have steak, you'll eat steak, but if you put spices and A1 sauce, you're actually opsonizing your steak and it makes it more appealing. Okay, so that's what cytokines do. It opsonizes the target invader. In this case, bacteria is the invader. That's what cytokines do. Opsonize the bacteria. It's also like, in comparison, it's like painting a target with a laser so that a missile knows where to strike, like guided missiles. So that's why, you got pinpoint accuracy of the drones and guided missiles. A laser will be pointed to that target, and that's where the missile will know where to go. So cytokines do that. It's like painting a target with a laser so that a missile knows where to strike, and that's how you get pinpoint accuracy, and that's just a comparison. So cytokines enable the migration of leukocytes to the site of inflammation or infection in interstitial tissue. So what cytokines do? It enables the migration of white blood cells. And this is diapedesis. So here you got cells. This is the diapedesis where these cells will actually seek through the cell walls of capillaries. When you see this is called diapedesis. So they want to go to the site of injury so bad that they'll cheat. And by cheating, they'll have the diapedesis. Okay, this is diapedesis. Okay, interleukins. Interleukins are a group of cytokines. Interleukins are a group of cytokines that were first seen to be expressed by the white blood cells. Okay, interleukins derived by inter as a means of communication. So here we're still talking about communication, deriving from the fact that many of these proteins are produced by leukocytes and act on leukocytes, interleukins. Again, communication between white blood cells. Okay, just like cytokines. So interleukins basically is just a group of cytokines. Okay, I'm going to go through various types of interleukins, but I'm not going to ask you what is IL-1, what is IL-5, and what's IL-7. This is just to give you an idea of the different types of interleukins that are out there. So interleukin 1 is responsible for the stimulation of T cells and antigen presenting cells. And I'm going to talk about antigen presenting cells shortly. B cell growth and antibody production. That's B cells. Okay, those two go hand in hand. B cell growth and antibody production. IL-2 is responsible for the proliferation of activated T cells. IL-3 is responsible for the growth of blood cell precursors. So as you know, blood cell precursors are found in the bone marrow. So IL-3 is active in the bone marrow. IL-4 is B cell proliferation and IgE production. Okay, IgE production. IL-7, thymus and bone marrow stromal connective tissue cells. Okay, thymus and bone marrow stromal connective tissue cells. IL-9 promotes the growth of T cells and mast cells. IL-10 will inhibit inflammatory and immune response. Why do you think that would be good? Why do you think it would be important to have IL-10? It will inhibit inflammatory and immune response. Why do you think it's important to have that?
Is it for for our autoimmune?
Autoimmune? No, it's a little more simpler than that. So say for example, you get a cut, what's going to happen?
Inflammation.
You're activating, when you get a cut, your body's going to activate what? It's going to activate your inflammatory and immune response, right? And what's going to happen? All your white blood cells and all your proteins and are going to, and a lot of proteins are going to be activated and sent to that site of injury, correct? That's your inflammatory response. But why would you need IL-10? It inhibits the inflammatory and immune response.
To turn off the response?
Exactly. You need to turn off the response. So what's going to happen if you didn't turn off the response? Your arm's going to be swollen up like Popeye because all the white blood cells in your body are going to be deployed to that site of injury. You don't want that. So you need to have IL-10 to turn off the inflammatory response. For every response that you have, there has to be a balance and a mechanism for turning it off, because you don't want to, you turn on the faucet and you just don't want to walk away from it. You have to turn it off. Okay, so good. IL-16 is chemo attracts CD4 T cells. I'll talk about CD4 T cells coming up. IL-17 activates the memory T cells. Interferons. Interferons are proteins made and released by host cells. Okay, a host cell is any kind of cell. I mean, like a cell lining your lung can be a host cell. Okay, the cells lining your lung, there's cells lining your stomach can be a host cell because they can be infected in response to the presence of pathogens such as viruses, bacteria, parasites, and tumor cells. Those pathogens will infect what's called a host cell, especially viruses. If you get a cold and you get viral pneumonia, the viruses have invaded cells in your respiratory tract. Okay, that's what host cells are, and those are the pathogens: viruses, bacteria, parasites, and tumor cells. They bind to receptors on the target cells. The target cells or the host cells. Okay, so the viruses, bacteria, parasites, and tumor cells will bind to certain receptors on the target cells. They're going to target cells of your lining your respiratory tract or your stomach or whatever. Okay, the various host cells. An interferon allows communication between the cells that trigger the protective defenses of the immune system to eradicate pathogens or tumors.
Professor Badger, let me interrupt for a second.
Pardon me?
I'm sorry, let me interrupt for a second. I have a weird question here for you. When you were talking about about the interferons, you mentioned that they bind to target cells. We're not talking about the actual target cells, the ones that look like targets. We're just talking about
the target cell can be a host cell.
Okay.
Say, for example, I don't know any kind of virus, but a virus that'll cause you to have viral pneumonia. Okay, the target cell would be a respiratory, a cell that's lining your respiratory tract. That could be like a target cell. So the virus will attack that, and then you end up having viral pneumonia.
Perfect. Thank you very much.
You're welcome. Okay, so interferons again allow communication between the cells. So there's communication between the cells, and it triggers the protective defenses. So again, there's trigger the protective defenses. All of this is about how your body is responding to some kind of stimulus or infection. When that happens, when you have a stimulus or infection introduced to your body, there's a whole bunch of mechanisms that take place, and coagulation, activation of lymphocytes, the interferons, the cytokines get activated, a whole myriad of things that take place, and it's in communication between the cells is imperative. So here, interferons allow communication between the cells. Communication between the cells. That's the crux of the matter. Okay. All interferons share common effects, and they are antiviral agents, and they can fight tumors. So interferons can be specific for fighting viral infections, and also can be used to treat tumors, cancer. Okay, so interferon therapy, for example, if a patient has chronic hepatitis, I understand that chronic interferon therapy can be used for patients who have chronic hepatitis that has activated too, and you don't want it to become fulminating hepatitis. Once it becomes fulminating hepatitis, then you probably past the point of no return, and the disease has progressed to a pretty bad stage, or tumors. You don't want it to tumors to progress into bad terminal cancer. So interferon therapy is really important for viral infections and for treating cancer. Okay. They're part of the non-specific immune system and are induced at an early stage in viral infection. And that's an important point because catching a virus infection at an early stage is really important to prevent further infection. So keep that in the back of your mind. Early stage of viral infection, identifying the early stage is really important. Okay. At the surface of each T cell displays thousands of identical T cell receptors. T cell receptors on the surface of each T cell. And it binds to a T cell, a T cell receptor binds to a bimolecular complex displayed at the surface of an antigen presenting cell. So an antigen presenting cell is a cell that's really important in presenting an antigen. Say, for example, a cell is infected. A cell is infected by a virus. Okay, that cell is doomed. That cell is going to die and it's going to be destroyed. Okay, so, but that's also an antigen presenting cell. What it does is that portions or parts of that virus or antigen that has infected that cell gets exposed. It'll expose itself to the surface of the cell. Okay, so somehow it'll leak what's infecting it to the surface of the cell. And what happens is remember the cytotoxic T lymphocytes? It's floating around in your bloodstream and it's looking for the pathogens. So an antigen presenting cell is actually presenting a portion of the antigen that has infected that cell. And once it sees that, then it'll allow the cytotoxic T cell to swoop down and destroy that cell. So APCs or antigen presenting cells displace antigens complex with MHCs. MHCs are the major histocompatibility complexes. It just makes the antigen more visible. So in previous classes, the comparison I made, and please don't laugh, it's like, you know, in the movie Lion King. Remember how Rafiki went up to Pride Rock and he raised, I guess it's Simba, up in the air. I guess he's presenting Simba to the gods, right? So I compare that to the antigen presenting cell. But we're not talking about anything swooping down and trying to gobble up Rafiki and Simba. But anyway, that's the antigen presenting cell. It displays the antigens that has infected that cell that's complex with a major histocompatibility complex. Okay, I'll get back to that infection of this host cells later. Most of the T cells in the body belong to one of two subsets, the CD4 and the CD8. And these are distinguished by the presence of glycoproteins on their surface. CD8 T cells are bind to epitopes that are part of the class 1 histocompatibility molecules. Just remember, CD8 is class 1. Almost all the cells in the body express class 1 histocompatibility molecules. I may ask that question on a quiz. CD8, all cells of the body, I could ask the question, all cells of the body express class 1 histocompatibility molecules. What type of T cell am I talking about? And it's either going to be CD4 or CD8. CD4 cells bind epitopes that are part of class 2, and only specialized antigen presenting cells express class 2 molecules. Okay, and this is important because dendritic cells are like dendrites. Dendrites are cells of the nervous system, and these cells process antigen material and present it on the surface cell to the T cells. So that's what I was talking about. An antigen presenting cell, like a dendritic cell, a dendritic cell is an antigen presenting cell. Okay, So they process antigen material that that host cell has been infected with and presents it on the surface of the cell. That's what a dendritic cell is, and it's an antigen presenting cell. Okay, so that's how it's what it's doing is that cell is doomed, but it's going to get rid of the virus by exposing or presenting it to the surface of the cell so that the T cells can identify it and eventually destroy it. Okay, CD8 T cells are cytotoxic T lymphocytes. Remember, those are the ones that are floating around, and they look for cells that have presented antigens that have infected cells. So this is the CD8 is a very useful function if the target cell is infected with a virus, because the cell is usually destroyed before it can release the fresh crop of viruses able to infect other cells. And that's why timing is critical. You want to catch it before it's able to infect other cells. Okay, you want to catch it when at an early stage of infection. Okay, the target cell is infected with virus. That's a given. But if nothing happens, and what will happen is inside the host cell, the virus will replicate. It'll burst. There goes the host cell. The viruses will now be free to migrate during a short period of time and infect other host cells. Okay, you need to during that time, the viruses are vulnerable. So that's why it's critical for it to get to another host cell and start replicating again. So every time you get a virus infection, for example, influenza virus, the virus will invade certain cells of, let's say, of your respiratory system, okay? In the case of respiratory passages. Once inside the cells, once the virus is inside your cell, it's safe. More, and then it'll replicate and it'll make more viruses, okay? It'll replicate more viruses, more pathogens. Okay, while it's inside the cell, it's safe because it's not exposed. So different macromolecules are made and encoded for the viral genome. That's basically replication. So in due course, these are assembled into a fresh crop of virus particles. This, again, this is within the host cell. And then what happens is, once it gets to a point, it's going to leave the cell, often killing the cell in the process, and spread to new target cells. So the cell is going to burst, and immediately it's going to swim or move to another host cell and infect it. Except when in transit from their old home to their new home, the viruses work inside of your cell. So once it's inside a cell, it's safe from any antibodies that can potentially invade it. But once it's between an old cell to a new cell, then it's vulnerable. But once it's inside a cell, it's safe. So early in the infection process, what will happen is, okay, early in the infection process, the infected cell, that host cell, will display fragments of the viral protein. Remember that the viruses are going to replicate and make more viruses. Well, the cell is actually getting back to the virus that infected it. And it does so by exposing some of the fragments of the viral proteins to the exterior of the cell. The infected cells will display fragments of the viral proteins in the surface of the class 1 molecules. So now the viruses are exposed. So CTLs, or cytotoxic T lymphocytes, they are specific for that antigen. And once those viral proteins are exposed, it'll be able to bind to the infected cell and often will be able to destroy it before it releases a fresh crop of viruses. And that's why timing is critical. That's why it's important to get these cytotoxic T lymphocytes to attack these cells at an early stage, okay? You want to destroy the infected host cell before it can release a fresh crop of viruses. And the cytotoxic T lymphocytes know which cells have been infected by viruses by the antigen presenting cells displaying the fragments of the viral protein. Okay, you guys understand that process? If if it's not for the antigen presenting cells to display the fragments of the viral protein, the cytotoxic T lymphocytes will not know which cell to attack. Okay. So in general, the role of the CD8 T cells is to monitor all cells of the body, ready to destroy any cell that express foreign antigen fragments. And that's the important part. Cells that express foreign antigen fragments, the cytotoxic T cells will identify those cells for destruction, okay, in their class 1 molecules. Basically, the CD4 cell is a co-receptor, and it helps out the T cell receptor with an antigen presenting cell. It's not an antigen presenting cell, but it just helps out the antigen presenting cell. Okay, it's a co-receptor. Next is a cluster of differentiation. It's just a designation, and it's often abbreviated as CD. Kind of like the IL. I'm not going to ask you too much on the IL. The CD, similar. It's just a classification. It's used for the identification and investigation of cell surface molecules. CD molecules act in numerous ways. They act as receptors, act as ligands. A signal cascade is usually initiated, altering the behavior of the cell. If you can notice, there's nothing red on this page. Same with this one. So the different types of CD markers, you got stem cells, granulocytes, like the cells of the myeloid system. It has a CD45 CD marker. T helper cell, cytotoxic T cell, those are lymphocytes, and the B lymphocytes. They have different CD markers, and a natural killer cell. Those are all the CD markers of those different types of cells. Okay, the CD4 level, this is what doctors need to identify the type of viral infection that you have. So a normal CD4 count in a healthy HIV negative, an HIV negative adult can vary, but is usually between 600 and 1200 CD4 cells per cubic millimeter, and it measures this by flow cytometry. And like I said, it's monitoring. That's why it's important to know how to calculate the absolute count of lymphocytes, of neutrophils. Okay, you know how to calculate absolute counts. So normally, in an HIV negative patient, it's between 600 and 1200 CD4 cells per cubic millimeter. It's done by flow cytometry. The doctors want to assess the number of CD4 cells in the lymphocyte population, again, the absolute count. And this is called the CD4 percentage. In HIV negative people, the normal result is around 40%. A CD4 percentage, which falls below 15%, is understood to reflect a risk of serious viral infection. Okay, so the doctors are looking for really low CD4 counts. Again, this is why they order flow cytometry. It's also known that CD4 cells differ in different populations. For example, the Chinese and North Indian normally have a lower CD4 count than people in Western countries. So just because people of Chinese or Oriental or North Indian origin, they have a CD4 count, doesn't necessarily mean they're walking around with virus infections. That's their demographic, okay? All right, pictures, the picture portion of this lecture. So that's a normal lymphocyte. This is a normal lymphocyte. This is, if you recall the blast of the myeloblast, this is coarse clumped. This is a mature lymphocyte. See how dark and clumpy it is? And then don't think that this is a high NC ratio because there's not too much cytoplasm. But generally, the lymphocyte is approximately the same size. I know you're going to call me a liar. Well, it's not approximately the same size of a red blood cell, but usually lymphocytes are compared to red blood cells. This one, okay, normal, round, not round, but it's oval nucleus, no folding. It's coarse and clumped. And if you look at the cytoplasm, it's bluish. Okay, it's bluish. So this is a normal lymphocyte. It's oval, no folding, it's coarse clumped, and the cytoplasm is bluish, okay? Here again, another normal lymphocyte, no folding. If you can see the clumping of the chromatin, this nuclear material is called chromatin. And then you have cytoplasm here. And then you can see that there's bluish tinging on the edges of the cytoplasm. That's a normal lymphocyte. No folding, coarse clumped nucleus. Normal lymphocytes again, roundish. This one is almost, I mean, kind of the same size as the red blood cell. It's roundish, no folding, dark, coarse clumped, thin cytoplasm. It's kind of light blue here too. No folding of the chromatin. It's dark and it's coarse clumped. Okay. Here, this is a lymphocyte. Okay, with pseudopods. This means this lymphocyte is active. It's moving around throughout your blood. It's looking for things. Okay, this could be like a cytotoxic T lymphocyte. It's a reactive lymph. Okay, you see the bluish tinge. If I see bluish tinges like that, it's probably a lymphocyte. But here it's larger. It's not a blast. It's mature. The chromatin is, again, it's kind of clumpy. It's not fine. Remember, the early immature stages, the chromatin is fine. But definitely, this is not a blast because blasts don't have pseudopods like this. And then also too, the nucleus of something that immature is not this coarse clumped, okay? So this is a reactive lymphocyte or a reactive or slash atypical. I will accept reactive slash or atypical, okay? More reactive lymphocytes, look like these lymphocytes are moving around. It could be like cytotoxic T lymphocytes looking for stuff, okay? It's not round, but these are lymphocytes. You can hardly see the cytoplasm, but if you see this, it's mature as opposed to immature. An immature cell will not have this kind of shape. An immature cell would be more oval or round shaped, but this is definitely mature. It's moving around. It's starting to look for things. So these are reactive lymphocytes. Atypical lymphocyte on the left and a reactive lymphocyte on the right. See the pseudopods on the reactive lymphocyte. This is an atypical lymphocyte. This could also be what's called plasmacytoid. And then I'll get into what plasmacytoid means because it has this little huff here. An eccentric nucleus with a huff here is almost plasmacytoid. But that is an atypical lymphocyte. This is an atypical lymphocyte. I will use, for this class, I'll use atypical and reactive lymphocyte synonymously, okay? I'm not gonna mark you if you say reactive. No, that's atypical. The argument, there's not gonna be an argument. It's either atypical and it can also be reactive.
Professor, I had a quick question before you move on.
Okay.
So what would be the difference between that reactive lymphocyte and a monocyte? Because they kind of look the same.
Okay, I have a whole PowerPoint presentation because that's a good point. A lot of classes, even at the CLS level, have trouble distinguishing between a monocyte and a lymphocyte. Do you know what a monocyte looks like?
Yeah, it looks like that, but it has a smaller nucleus.
And where did you learn this? Not from me, because my next lecture is monocytes.
When we were doing those white blood cell counts in John's class, I had a lot of lymphocytes, and I asked him about it, and he was saying that
They're monocytes.
I was probably counting my monocytes as lymphocytes.
Okay, I will tell you the difference between, because I have a whole exercise of and it's called lymphocyte versus monocyte. Okay, so you're going to go through a whole bunch of slides, and it's going to ask you lymphocyte or monocyte, and then you'll say lymphocyte, and you'll say lymphocyte, and you'll say monocyte. Okay, so it'll become clear to you what a monocyte looks like. There's specific characteristics of monocytes that are not found in lymphocytes. Like I kept saying, no folding here, right? No folding here, right? And coarse clump chromatin. Whenever describe the chromatin pattern of a lymphocyte. There's no folding, there's no bending, and the chromatin pattern is usually coarse clump. Okay, but thanks for the question, because the next lecture is monocytes. Atypical lymphocyte. Now this almost looks like this a little bit. This one has more pseudopods, but this is an atypical lymphocyte. Atypical lymphocyte has what's called, this is skirting. This is a lymphocyte. Again, look at the nucleus. There's no folding and it's coarse clumped. Wait till we get into the monocytes. There's folding, and then the nucleus or the chromatin pattern is lacy. Lacy meaning it's kind of spongy looking, okay? This is not spongy looking, it's dark, it's coarse clump. This is an atypical lymphocyte because it has a lot of cytoplasm. You see that? And this is called skirting. Atypical lymphocyte, to make note of this, what I'm going to say is if you report atypical lymphocytes, the doctor's going to want to know if you're seeing atypical lymphocytes, because atypical lymphocytes indicates a possibility of infectious mononucleus infection. Okay, atypical lymphocytes. So if you have atypical lymphocyte and infectious mononucleosis infection, you guys know what antibody would be, what virus would be positive? Virus level would be positive. That would be the Epstein-Barr virus. Have you heard of that? Okay, Epstein-Barr virus, atypical lymphocytes, infectious mono. Okay, infectious mono, atypical lymphocytes. So atypical lymphocytes are reportable. And if you report atypical lymphocytes, the doctors will clue on whether or not, if you report atypical lymphocytes, it's going to turn on a light in his head, and it's going to make him think in the direction of infectious mononucleosis. So this is atypical lymphocyte because of the amount of skirting. Look at the cytoplasm here around the nucleus. That's called skirting. And this is definitely, it's not a monocyte. If you look at the chromatin, there's no folding. It's not spongy looking. It's kind of coarse clumped. When we get into the monocytes, I'll point that out, what that looks like a monocyte. All right, plasmacytoid. Plasmacytoid means anything you see plasmacytoid means you don't have enough confidence to call it a plasma cell, so you call it plasmacytoid. Also, going back to last Wednesday's lecture, Pelger-Hewitt, remember Pelger-Hewitt, the bilobe neutrophils, the mature neutrophils that are bilobed, that's called Pelger-Hewitt, correct? Well, if you're not confident in calling it Pelger-Hewitt anomaly, then you can just go ahead and report it as Pelgroid neutrophils. Pelgroid. So Pelgroid means Pelger-Hewitt-like. Plasmacytoid means plasma cell-like. So this cell here is, I would have enough confidence to call this a plasma cell. Plasma cell, by definition, is round nucleus. It's eccentric, meaning it's not in the center. And then it has this little huff here of Golgi. You see that, just like the Golgi in the myelocytic series, the myelocyte, right? Okay, so this is a lymphocyte. This is a lymphocyte. It's not a myelocyte because this is pretty blue. Myelocyte is not, one does not have this much nucleus. I know throwing all these cells at you can be pretty confusing, but this is a plasma cell, really round nucleus, eccentric. It's off to the side. It has this huff here. That's the Golgi, and blue cytoplasm. So this is lymphocytic. If you see the blue cytoplasm, it's lymphocytic. If you round nucleus, coarse clump, it's a plasmacytoid. It's eccentric. So that's a plasmacytoid lymphocyte. These two cells here, if you can recall, are NRBCs. Okay, memorize the cytoplasm. Perfectly round, ground glass cytoplasm, gray ground cytoplasm. Those are nucleated red blood cells. This is plasmacytoid again. It's large, blue cytoplasm. Blue cytoplasm puts it in the lymphocyte category. If you see this much blue cytoplasm, you put it in the lymphocyte category. So for the white blood cells, you have your myeloid series that leads to neutrophils. You have your lymphocyte series, which we're talking about now, and then you have your monocytic series. You don't see any granules here, but you see a lot of blue. If you see a lot of blue, that's probably a lymphocyte. So lymphocyte versus monocyte for distinction. Okay, monocyte cytoplasms are not this blue. Okay, so you're not ever going to call this a monocyte. This here, that could either be that could be a monocyte, but look at it's pretty granulocytic. It could be a band, but it can be monocytic. So I'm not going to focus on that. This is just focus on this one. This one almost is the color of a monocyte, but I'm not gonna talk about that cell. So large cell, it's lymphocytic, but see the blue cytoplasm? When you see blue cytoplasm like that, it's lymphocytic. When you see the huff here and eccentric nucleus, it's plasmacytoid, okay? Meaning it's a plasma cell, but it's probably like a plasma cell. Again, another plasmacytoid, eccentric nucleus, blue cytoplasm, and a little bit of a huff here. Again, the nucleus is coarse clump and no folding, and it's not spongy looking. It's not spongy looking, and I'll point at the sponginess of a monocyte. Okay, these are perfect plasma cells. Eccentric nucleus, perfectly round. You got a huff here, the Golgi. Okay, these are plasma cells. I kind of, the comparison I like to make of these are, they look like deviled eggs. Okay, I don't know why I use food analogies, but to me, they look like deviled eggs. If you've ever had deviled eggs before. All right, eccentric nucleus, huff, and bluish. It's like an egg shape. I mean, they're egg shaped, you know. These are perfect plasma cells. Plasma cell, egg shape, eccentric nucleus, the huff, that's a plasma cell. You see that? And if you notice, excuse me, the chromatin is perfectly round. See, they're perfectly round. There's a lot of things that are perfectly round. Howell Jolly bodies are perfectly round. They're perfectly round marbles, as opposed to maybe like a platelet artifact. Howell Jolly bodies are perfectly round. Nucleus of a plasma cell is perfectly round. Where are they? NRBCs, they're perfectly round, okay? So there are a few things that if you see are perfectly round, and there's not that many of them, you need to say, yeah, it's either an NRBC, it's either a Howell Jolly body, or it's either a plasma cell. Plasma cell, perfectly round, eccentric, Golgi huff, and shaped like an egg, deviled egg. Another plasma cell. I hope you're getting the point. Eccentric nucleus, perfectly round, blue cytoplasm, oval shape, shaped like an egg. Okay, that's it for lymphocytes. Are there any questions? That's a lot of information, especially with the different types of lymphocytes. Remember, don't focus too much on the interleukins and the cluster differentiation. I'm not going to ask you. If I see one, I won't mark it on your quiz. All right. Let's take, you want to take 10 before we go to monocytes?
Yes.
All right, I'll see you guys at 5:20.